Hi, dear friends, cultural creators. Bruce here with a story about history, my history. In fact, 50 years ago, my life changed when a radical experiment revealed something that wasn't even in any of the biology books or the curriculum that I was involved with in graduate school. At the time, I was helping students with an understanding about genetics and how genes control our life, the belief in genetic determinism that at the moment of conception, the genes that we get are a blueprint of the life that we will lead on this planet. Now, the problem with that, of course, that makes us victims of our heredity because since, as far as we know, we didn't pick the genes we came with, can't change the genes if we don't like the characteristics, and these genes control us, then by definition, we're victims. And while I was teaching that, I was also working on stem cells in my laboratory. And the stem cells are, well, what is a stem cell? It's an embryonic cell. Why should I call it a stem cell? And the answer is once you're born, you still have embryonic cells in your body, but you're not an embryo, so we just changed the name. But it's a multipotential embryonic cell. So the next question is, why have stem cells? And the answer, again, is a very simple answer. Because the human body is made out of a community of 50 trillion cells. And every day, we're losing hundreds and hundreds of billions of cells every day. Just normal attrition. Skin cells keep fluffing off every day. The lining of the digestive tract, the entire lining of cells from mouth to anus is replaced every 3 days. That's nearly 1 trillion cells. Well, if we're losing cells every day, then darn it, we must be making some cells because how many days in a row can you keep losing cells without replacing them? So if you're listening to this and you understand what I'm talking about, then you already have stem cells. For the simple reason is if you didn't have stem cells, you wouldn't be here. So all of us that are here right now in this community, listening and talking, we all have stem cells. Now they're multipotential cells. Now what's interesting is 50 years ago, I was cloning stem cells, and I put a single stem cell in a culture dish by itself. The cells would double the population every 10 or 12 hours. So first there's 1 cell, then 2, 4, 8, 16, 32, double, 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 double. And in 1 week, I have about 30,000 cells in a petri dish. And most important point, they all came from one parent. So I have 30,000 genetically identical cells. I split those 30,000 cells into 3 petri dishes, and I changed the culture medium. That's the environment that cells grow in. Culture medium is the laboratory equivalent of blood. So when I grow human cells, I look at what human blood is made out of, make a composition in the laboratory, call it growth medium. But since I'm making a laboratory, I can adjust some of the parameters. So in this particular time, I created 3 different versions of growth medium, slightly changing the chemistry in each one. And now I have 3 dishes with genetically identical cells in all 3 dishes. And I feed each dish a different culture medium, a different environment. The result was in dish 1 with environment A, cells form muscle. In dish 2 with environment B, the cells form bone. And in dish 3 with environment C, the cells form fat cells. What really got me was the fact that all the cells were genetically identical. So the fate that the cells express were not based on the genes, but it turns out to be based on the environment. Well, this blew me away because all of a sudden I started to recognize environment was controlling the genes and regulating genetic activity. And yet, there I am in a course teaching a curriculum that genes control life. And when I tried to show it to my colleagues, they looked at it, and since they were so caught up in genes and the whole story of genetic determinism, they looked at my work as something artifactual, can't be real, it's not relevant, and they dismissed it. And the primary reason they dismissed it was simply because they said, okay, Bruce, you've changed the environment and the genetics changed. Tell us how that happens. Because remember, these people in genetics were already involved with genes turning on and off and regulating their own activity. And now, where does the environment come in to actually regulate the genes? I had no answer. So basically, they ignored me. They saw the work. It was repeatable every day. But I couldn't come up with how the environment was translated into biology. So I started to look at the cell from a point of view of how does an environmental signal affect genes, and start to physically look, where does an environmental signal even come into the cell? Well, the first thing is the cell has a skin, and it's called the cell membrane. the cell membrane at that time in conventional biology was equated to something like saran wrap, plastic wrap. The membrane of the cell, that very thin layer that you couldn't even see in a regular microscope, it could only be seen in an electron microscope, the membrane was so thin that they just attributed the character to its simple structure as being nothing more than a plastic wrap to hold the cytoplasm together. But as I started to focus on the structure of the cell membrane, I saw there were things that they really weren't talking about in the context of membrane function. And that is that there were two kinds of proteins built into that membrane. One protein had an antenna on it, and it's called a receptor. A receptor is a protein that reads an environmental signal. Connected to the receptor is an output device, a device that creates a response when the receptor sees a signal. And the main class of these output proteins are called channels. And basically, a channel is a protein that is like a solid ball, but when the signal activates the channel opens up and makes a conduit between the inside and the outside of the cell. So I'm working on the cell membrane and the dynamics and trying to figure out how that environmental signal gets into that nucleus and affects it. And so I started to define the membrane in terms of its physical structure, a definition that was relatively novel because it was based on the crystalline structure and the protein composition. And here's the point. A membrane basic structure is a bimolecular layer of phospholipids. Well, man, that sounds very scientific. What is a phospholipid? Well, that's a molecule that has a lipid or fat component as well as a water-loving component. And why is that relevant? Because fat excludes water and water-loving elements absorb water. So when you look at the crystalline structure of these phospholipids built into the membrane, it appears from the distance as a three-layered structure in the electron microscope, dark, light, dark. The middle light layer in that membrane is where the lipid molecules are. An analogy is a membrane is like a bread and butter sandwich, that the outer portions of the membrane, the phospho groups, actually are the bread part, and remember, bread soaks up water. But in between the phosphate groups is the lipid, the butter part, the fat part. That separates the inside of the cell from the outside of the cell. In other words, if something's in the environment and it's like a drug in the water environment, can that drug get into the cell through the membrane? The answer is no. Why? Because while the drug may be absorbed on the surface of the membrane, where the phosphorus is absorbing water, the drug cannot pass through the lipid, the butter part of the membrane. So by definition, the membrane is a non-conductor. Why? Things in fluid, such as nutrition, such as the hormones, the emotional chemicals, these elements are dissolved in water and cannot easily penetrate a membrane. As a matter of fact, the lipid in the center of that membrane, dark, light, dark, the lipid actually repels the water. So a membrane is a barrier. It's a non-conductor. Well, the problem is if the membrane's a non-conductor, then how does food get in the cell? How does waste get out of the cell? It has to pass through the membrane. So we start to find that there are proteins like channels. Channels, as I said, are the proteins like a globular protein that's closed, but when a signal hits that channel, it opens up and makes a tube that connects the inside and the outside. So stuff from the outside can go in the cell, or stuff from inside the cell can go out through the channel. But here's the important point about a channel. There are different kinds of channels, but the channels are specific to what they allow in and out of the cell. There are some channels that let potassium in and out of the cell. There are some channels that let calcium in and out of the cell. There's some channels that let hydrogen in and out of the cell. Some channels that let glucose in and out of the cells. When there are no proteins in the membrane, as I said, that bread and butter sandwich is a non-conductor. But once you put the channels inside the membrane, then things can be conducted across. Question? Can everything be conducted across? No. Only what the channel allows. So, the membrane with channels is not a non-conductor, because things can get through. But it's not a full conductor because it doesn't let everything through. So by definition, the membrane is called a semiconductor. And the two proteins that control what goes in and out of the cell are A, the receptor, which receives a signal, and B, the channel, which opens up and lets the response go through the cell. The receptor is an input. The channel is an output. The point is simple. Built into the membrane are units called receptors and channels that represents units of information. Now, when we talk about an input and an output, we can also use the term a bit of data, because in an information processor, a bit of data represents a gate and a channel. But a gate is another word for receptor. So I sit down at my computer and I'm typing on the computer the characteristics of the cell membrane. And the characteristic I defined at this moment in time in 1985 is the following. The membrane is a liquid crystal semiconductor with gates and channels. And in 1985, as soon as I wrote that down, I said, my God, I read something like that somewhere. And there it was, right next to my first Macintosh computer, a book from Radio Shack, the Simpleton book, Understanding Your Microprocessor. And there it was right in the introduction. The definition of a computer chip is a crystal semiconductor with gates and channels. When I read the definition in that book and recognized it was the same definition that I just described for the cell membrane, at first it was an exciting moment. Wow, what a coincidence. The definition of a computer chip and the definition of the cell membrane are exactly the same. But as I started to look at it, I started to recognize that every identified part in the silicon-based computer chip that we find in our digital world, the same parts are present in a cell membrane. The difference is the computer chip is silicon-based. The cell membrane is a carbon-based chip. And yet they both exactly have the same function and the same mechanisms concerning receptors and channels and the ability to transmit information across the crystal. It's all exactly the same. Oh my God. The cell membrane is an information processor. It reads the environment through the receptors and translates that information into the cell via the channels, and the channel activity controls functions inside the cell. And it's like, oh my God. I saw it and I said, oh, a cell is a chip. And the nucleus with the DNA are programs in it. And while in the classroom I was teaching that the DNA was the equivalent of read-only memory, meaning whatever your gene is, that is your life, hence the nature that genes are blueprints of our life, it turns out in the biology, the nucleus is a disk with programs, but it's not read-only. It's read-write, meaning environmental information translated by the cell membrane, the information processor, releases information into the cell that selects the gene programs, but not only selects the gene programs, but can rewrite the gene programs. It was at that moment that I recognized 50 years ago the nature of what we refer to today as epigenetics. Genetics and genetic determinism, which I was teaching medical students, genetic determinism simply means gene control by genes. But the new science is called epigenetic control. Epi means above. So the new science is basically control above the genes. And then I said, oh my God, the membrane is that structure above the genes that controls the activity of the genes. And since the membrane is reading environment, then by changing the environment of the cell, as I did in my tissue culture experiment, I elicited different genetic programs. And why was this so amazing was, well, in genetic determinism, the original belief, you are a victim of your heredity. You didn't pick the genes, you can't change them, and they control you. But in the new science that I observed was that I can change the activity of the genes via the membrane chip, which was responding to the environment, and I could rewrite the program. And the difference was what I was teaching was victimization. Genes control you, you do not control the genes. What my research on stem cells revealed is empowerment. It says that the genes are controlled by the environment and our perception of the environment as mediated by the cell membrane. I say, why is it relevant? Because I can change the environment. I can change my perception. And in doing so, I change my genetics. And all of a sudden it says, then I'm not a victim of my genes. I am the master of my genes. Why? Because the cell membrane, as an information processing chip, organizes the individual cell as a programmable chip. That's why a liver cell, a muscle cell, a skin cell, a bone cell, all have the same nucleus with all the same programs, but each one is individually programmable by the environment. And then the explanation of my research fully encapsulated the understanding and actually went far above the conventional world of genetics that my colleagues were into. Yet, it's still a hard drive to get them to move that way, and it took another 20 years before science has come around to fully recognize the nature of epigenetics and the power that it is. And the interesting aspect for me is, once you understand that the cell membrane is an information processor, once you understand that a receptor channel complex is a bit of data, you are given a vision of evolution that has never been seen before. Because up to now, evolution is based on presumably genetics. As you go up what is called the evolutionary tree, a model of a tree where bacteria, the lowest organisms, were at the base of the tree, and as you go up the tree, there are more and more and more complex organisms until you get to humans at the top of the tree, and you say, the tree of life. And scientists said, well, what is it that makes a difference between the bacteria and the higher forms? And the answer at that time was genes. That as you go up the tree, there are more complex organisms, and complexity was related to genes, so it led to a presumption that as you go up the evolutionary tree, there are more and more genes making more and more complex organisms. But that whole idea fell apart with the Human Genome Project, because it turned out that humans don't have any more genes than one of the simplest organisms, a worm, at the bottom of the tree. Humans have 20,000 genes, and this worm, with only 1,271 cells, has 20,000 genes. So the idea that genetic complexity was responsible for this tree of life turns out to be totally incorrect. A more correct interpretation was actually first provided by the first scientist to ever publish a paper on evolution, and that was Jean-Baptiste de Lamarck. Lamarck recognized evolution, pointed out the hierarchy of simple to more complex organisms, but he didn't know about genes, and yet he came up with a better understanding. He said it was the nervous system that was increasing as you go up the tree. So rather than measuring the evolution of an organism based on genes, it was measuring on the nervous system. Now again, there comes a problem, because in the conventional world, how does that nervous system work? Well, we're still lost in the whole idea of it, because a brain and a nervous system are very complex. And yet, a single cell has a nervous system. A single cell responds to the environment and can adjust its biology. So the relevance became simple. Instead of trying to understand the nervous system with a complex brain, try to understand the nervous system of a single cell. And guess what? The nervous system is a cell membrane. And the cell membrane reveals that it's a chip, and that the processing power of that chip is related to the bits of data, which are receptor channel complexes. And all of a sudden it says, oh my God, then evolution of the nervous system, the evolution of consciousness, is directly tied to the mathematics of how many bits of information, bits of data can an organism accommodate. And that's when all of a sudden the secret of life manifests, because it reveals that the receptor, effector, or channel complexes in the membrane can only monolayer. And that means the more bits of data you want to add to a membrane, the greater the surface area of that nervous system must be. And all of a sudden it was like, oh my God, for the first time in history, there's a physical measurement of consciousness and nervous system. It is how many of these bits of data, how many of these receptor channel complexes an organism can accommodate. And once we start to look at that, then evolution changes and we can understand the evolutionary tree of making more and more intelligence by making better and better chips, better membranes. And here we are, a human made out of 50 trillion programmable chips. Each chip can become anything: brain, skin, nervous system, muscle, but it's based on the environmental programming that determines the fate of those cells, and the membrane is that processor. So we move from genes to understanding how life evolved to looking at the cell membrane. And first time in the history of science, there is a mathematical foundation to plot evolution of life on this planet. And that is surface area rather than genes. And once you understand surface area, you can understand the nature of why our brains have so many folds, gyri and sulci, because inside our skull, to make more surface area of the brain, you have to fold it. And that is why when you go from lower vertebrates to higher vertebrates, the brain gets bigger, fills in the skull, and once the skull is filled in, the next level is to fold the brain into surface area, gyri and sulci, a reflection of surface area. The bigger the surface area of the brain, the more capacity there is to process information, the higher you are on the evolutionary tree. So what I saw 50 years ago seemed to be, to my colleagues, an artifact or just some silly part of the experiment, leads to the whole foundation of understanding the nature of life, how environment controls our biology, our behavior, and our genetics, and how evolution is derived from enhancing the information processing capability of our cells. More bits, more data, more awareness, more consciousness, more power. And that's where we stand today. 50 trillion cell computer, 50 trillion programmable chips called the human body. And the power that we have is all in that read-write genome that we can change and change our lives with through consciousness. Activating the bits, the receptors and the channels are how we control life.